Alrighty, today we are going to move into viral genetics. So we've talked about bacteria already, and now we're going to look at some viruses. You are actually going to start taking notes today. So up until this point, I've just been posting my entire PowerPoint, and starting today, you'll get a student copy of the PowerPoint, and anything that's written in blue on my slides will not be included in your regular notes. So those are things that you will need to type out, and then you'll be turning that into me as sort of a notebook check just so that I know you are paying attention and following along. The things you take notes on will also help you to answer the questions that we do at the end of the section. So we're going to start off with a video, and you guys have already done a little project with this type of stuff, so this should be a review.
Hi, my name is Paul Offit. I'm talking to you today from the Vaccine Education Center here at the Children's Hospital of Philadelphia. One thing that some parents wonder about is how does one make vaccines? Or said another way, what are the different types of vaccines? And here's what I would say. First, one way to make a vaccine is that you take a virus and you weaken it. Weaken it so that it can reproduce itself enough to induce an immune response, but not enough to cause disease. And that's the way that the measles vaccine is made, the mumps vaccine is made, the chickenpox vaccine, the rubella or German measles vaccine, and the rotavirus vaccines. Another way that you can make a vaccine is you can take a bacteria that normally makes a toxin, like tetanus or diphtheria. And so those bacterial infections are more intoxications, frankly, than they are infections. So you take the toxin made by those bacteria and you kill it with a chemical, and then that becomes your vaccine. That's the way the tetanus and diphtheria vaccines are made. And in some ways, the whooping cough vaccine is made similarly too, because the whooping cough bacteria also can make toxins, and so that is another example of that kind of vaccine. Another way you can make a vaccine is you can take a bacteria like the bacteria that causes a meningococcal infection, pneumococcal infection, another infection caused by Haemophilus influenza type B, and you take the complex sugar coating of that bacteria called polysaccharide, link it to a harmless protein, and then that becomes your vaccine. So another way, yet then the final way that one can make a vaccine is you can take a virus and instead of weakening it, you can kill it. And that's the way that the polio vaccine is made. That's the way that the rabies vaccine is made. That's the way the hepatitis A vaccine is made. And there's one more way that you can make a vaccine, which is that you can take just a part of the virus. So you just take one protein from the virus that you know induces an antibody response that can completely neutralize the virus, and that then becomes your vaccine. And those single protein vaccines are the hepatitis B vaccine and the human papillomavirus vaccine. Thank you.
So that's just a little refresher. You guys already had an assignment where you went through and actually told me about each type of vaccine. In case you don't remember, I'm going to review them really quickly now. So an inactive vaccine is also called a killed vaccine. It's created by inactivating the pathogen, meaning that it cannot replicate. Your body's going to recognize it as foreign and create antibodies against it. This type of vaccine will require booster shots, and the flu is this type of vaccine. An active vaccine is where you take the disease-causing bacteria or virus through a series of cell cultures, or animal embryos. Allow the virus to become used to replicating in a non-human cell until it loses the ability to replicate in a human. A lot of times they use chicken cells for this. You'll create a vaccine, provoke an immune response. The only bad thing about using an active one is that it can mutate and it can actually start reproducing again in human cells. So the idea is to remove it far enough away from a human cell to where it can't do these things, but we know that viruses, they don't have any checkpoints, and so they do mutate quite often. Obviously, we need a new flu shot every year. Toxoids. Some bacteria produce a toxin that can cause a disease. You're going to chemically or heat treat it to inactivate the toxin, and your vaccine would be created. This is what he talked about with tetanus. A conjugate vaccine. This was the last thing he talked about, where you have a piece of the germ, its protein or its caspid, which is the outer casing. It's going to be taken and made into a vaccine. It can be used on people with weakened immune systems, and for these ones, you'll also need booster shots. So they're safer if you need a booster shot, but you need more shots. So it's kind of up to you as to whether or not you and you want to go through 4 or 5 booster shots or just get the one, but you could potentially end up mutating it and end up with the disease. So you don't have any notes to take yet. There's nothing in blue. Everything's been in white so far. So TMV, this was the very first virus to be discovered, and it happened back in 1883. It's called the tobacco mosaic virus. And this was a plant virus. It damaged the leaves, flowers, and fruit, but it did not kill the plant. So I'll show you a quick clip of that, and then I think that'll be my last video.
Meet tobacco mosaic virus, TMV. TMV has a family called Vergiviridae. Like other plant pathogen viruses, pathogenic virus, TMV has a very wide host range. Symptoms that are induced by tobacco mosaic virus are somewhat dependent on the host plant and can include mosaic, mottling, necrosis, stunting, leaf curling, and yellowing of plant diseases. These symptoms develop quickly and are more pronounced on younger leaves. TMV infects at least 125 individual species including tomato, tobacco, cucumbers, pepper, and a number of ornamental flowers. Tobacco mosaic virus can be transmitted from the infected host to the healthy plants by human handling. Therefore, TMV can be transmitted from one plant to another by direct contact through its vectors, normally insects. In which, this can cause TMV to go through the infection process and then the replication process. The most common control methods for tobacco mosaic virus is sanitation, that would include removing infected plants and washing hands between each planting. TMV has a rod-like shape appearance. Its capsid is made from 2130 molecules of coat protein and 1 molecule of the genomic single-strand RNA that is 6,400 bases long. The coat protein assembles a rod-like helical structure around the RNA that protects the cellular enzymes by the coat protein. Fun fact: tobacco mosaic virus, the first virus to ever be discovered that damaged tobacco crops.
All right, so there's just a little history lesson for you. So the spread of the virus. Adolf Mayer determined that the disease was spread by spraying the infected sap from one plant onto another. Dmitry Ivanovsky filtered the sap and determined the disease-causing agent was not a bacterium. So up to this point, they were really used to just seeing these little bacteriums causing diseases. But the pores in the leaves were too small to allow bacterial cells through. So a virus is even smaller. Then they thought, well, maybe this is some type of a toxin. Maybe we were doing it to ourselves. Maybe it was a chemical toxin. So Martinez, I'm not even going to try to say that one, ruled this out by showing the plant could still transmit after many generations. So this toxin, if that's what it was staying inside of that plant for generation after generation after generation. Normally, toxins will dilute, but in this particular case, it wasn't diluting. It was actually becoming more powerful. Around this time, the first animal virus was discovered in cattle called foot and mouth disease, and that looks extremely painful for those poor cows. And then we came about with a human virus in 1900. This was the first human virus ever discovered. It was called yellow fever, and it was spread through mosquito bites. Mosquitoes do so much to us. It seems like they are constantly coming out with new diseases and viruses for us to catch. Symptoms included a fever, chills, muscle aches, eventually leading to organ failure, shock, bleeding, and death. A virus is a small infectious particle that consists of nucleic acid enclosed in a protein coat, and there are well over 4,000 different types of viruses. They're all pretty similar in that they have a small size and they need a living host in order to replicate. The differences between the viruses are the host range, so what can they infect? Can they go from people to animals, or are they simply just a plant virus? The structure of them, and the genome composition. So here's the first part that's in blue. Differences in host range. A host cell is a cell that is infected by a virus, and the host range is the number of species and cell types they can affect. So you'll want to write down that part in blue. The white part you don't need to worry about because that will be included for you in Canvas on the student PowerPoint. So you need to just have a Google Doc open, and that is what you will submit to me at the end of this lesson. Structural differences. Most are smaller than the wavelength of visible light. 20 to 400 nanometers in diameter, so they are extremely, extremely little. Over 50 million adenoviruses could fit into an average-sized human cell. So that is a lot of viruses that could get into one of our cells, and our cells are pretty darn small. But these are things like colds, sore throats, bronchitis, pneumonia, pink eye. Talked about a capsid. All viruses have a protein coat called a capsid. Again, this is in blue, so you're going to want to write this part down. Capsids are composed of one or several types of proteins and may be a variety of shapes. So I got a little guy here, and he is saying, if I coat myself with this protein, they'll never find me. And that's kind of what viruses do. Your body has a really hard time recognizing them, and a lot of times that's why the doctor will say, well, you just have to let it run its course. They can do things to help treat your symptoms, but they can't actually eradicate the virus from your cells. This is also kind of what makes the coronavirus so scary, is that we can't get rid of it. We can treat the symptoms, we think, but we're not really sure what the long-term effects of this virus are yet. And you can pause this video at any time if you need a little more time to write stuff down. A capsid is going to enable the virus to infect the host. Specialized proteins allow them to bind to cells. Bacteriophages or phages, these are viruses that infect only bacteria cells. They are a much more complex capsid for anchoring the virus to the host, and then they inject their viral DNA nucleic acid. So genome differences. A viral genome is a genetic material in a virus. It can be RNA or DNA, and it can be circular or linear. It can be single or double-stranded. So there's a lot of variables here with this genome. They vary in size from really tiny to fairly large, but nowhere near the size of our genome. It can contain a single genome or multiple genomes. So they have a lot of flexibility here. Viral reproductive cycle. So viruses do not act like living things. They're not made of cells. They cannot carry out reactions of their own. The only way for them to reproduce is if they get into a living host cell. So a virus's main goal is to infect a cell, and then it's going to make that cell produce new viruses. The first step is attachment. The virus must attach to the surface of a host cell. It's specific for one kind of cell because proteins in the virus recognize and bind to specific molecules on the cell surface. An example of this is HIV, which is the inactive form of AIDS. It has a spike glycoprotein that's going to bind to the protein receptors in the plasma membrane of human blood cells called T cells. And there's some movie out, came out a long time ago, and they talked all about T cells. Resident Evil is what that's called, and they talked all about a T cell virus, and a lot of people thought it might have been alluding to HIV, which does infect your T cells. So this is what HIV looks like. You see those little green guys? These are these tiny little probes, and they're going to stick to the cell. And all viruses have different types of hooks to dig themselves into your cells. Step 2 is entry. So the viral genome is going to enter the host cell, and then there's going to be a conformational change or a shape change for the phage to inject the DNA or RNA, whatever it has, into the host cell's cytoplasm. Step 3 is integration. For some viruses, this step is skipped. For others, they'll go through it. So integrase is an enzyme that cuts the host DNA, and it's going to insert the viral genome into that chromosome. Prophage. So the phage DNA is integrated into the bacterium. So with HIV, it's going to go through reverse transcriptase, which is a viral enzyme that's going to be released into the host cell along with the RNA. It's going to make a complementary strand of DNA, which your body does on its own. DNA is going to be inserted into the host cell's nucleus, and integrase is used to place it into the host chromosome. So this last bullet point here is in blue. Provirus is a viral DNA integrated into a eukaryotic cell. So that is how HIV actually digs itself into your cells, and it becomes part of your cell, which is one of those main reasons why your body doesn't fight against it, because it is part of your body. Step 4 is the synthesis of viral components. In bacteria, the host DNA is degraded, so it completely destroys everything inside of the bacteria. So that bacteria then only knows the virus. In eukaryotic cells like ourselves, the nucleus is going to produce many new copies of the viral RNA, and it's also going to start making viral proteins. It just isn't going to destroy our DNA. So our DNA is going to be made along with this viral DNA. Step 5 is viral assembly. In bacteria, phage components are assembled by proteins to make new phages. In eukaryotic cells, capsid proteins assemble spike glycoproteins during budding. So down here, this is where the new little phage is starting to come out. Most of the time, your cells aren't going to have these little spiked glycoproteins on them, but because it has this HIV viral DNA inside of it, the cell begins making it. And it's going to bud off, so it's going to pinch off here, and it's going to go off and infect other cells. Step 6 is the release. In bacteria, the phages are going to create lysosomes to digest the bacterial cell wall, causing the cell to burst. They're going to move on to infect other bacteria. In eukaryotic cells, budding is going to occur. So a piece of the host cell membrane is going to be studded with these viral glycoproteins, and it's going to leave the cell. Latency. Viruses can remain dormant for a period of time. Viral production is going to stop at step 3, and then eventually it'll pick back up again. So for example, HIV and herpes are both viruses, and they are both genital and oral. You can get them either way. And you don't become infected immediately. So you may not know that you have HIV or herpes for a couple of years or even a couple of months. It just depends on your body and your own stressors, what's going on, and that's normally whenever you'll have an outbreak of something. In bacteria, the cell is going to divide and copy the prophage DNA along with its own without producing a new phage. Newly arising viruses or emerging viruses lead to significant loss of human life, as we have seen with the coronavirus. New strains of influenza arise regularly due to new mutations. Viruses are highly volatile in that they are constantly changing. Now, the coronavirus is kind of interesting in that it wasn't randomly made up. It was more engineered whenever they were kind of messing around with different things, but I'm not going to get into that. If we just look at the flu, they are constantly updating that flu vaccine with what they believe will be the most prominent strain. They don't know for sure, and that flu virus is going to mutate again next year. SARS is an RNA virus, and this is a type of coronavirus. It's a severe acute respiratory syndrome. It mutated from viruses that infect humans and livestock. Human immunodeficiency virus. It starts with an acquired immune deficiency syndrome. So this is HIV, primarily spread by sexual contact, blood, and it can be passed from the mother to an unborn child. Worldwide, nearly 1 in every 100 adults between the ages of 15 and 49 are infected, which is crazy high numbers, I think. 40,000 new HIV infections occur each year. 70% are in men and 30% are in female. The destruction of the helper T cells. These are a type of white blood cell. The T cell facilitate the production of antibodies and other molecules that target and kill foreign invaders of the body. T cells are going to be destroyed by HIV, which means your immune system function is then compromised and you're susceptible to infectious diseases. So that's why people who start to get an active form of HIV normally get sick, and you can end up dying from like a common cold. Mutations. So HIV uses reverse transcriptase, which lacks this function. It's going to make many, many errors. There is no checkpoints. It creates mutant strains which become resistant to the antibody's defense, or to the body's defense, and it makes it difficult to create a vaccine, which is why we don't have one yet, but we do have treatments. Mutant strains become resistant to antiviral drugs. The death rates are falling due to the study of HIV life cycle. Antiviral drugs are being made to stop the proliferation of HIV cells, which means that those cells are not replicating. Protease inhibitors are going to prevent viral replication. A couple challenges is finding drugs that stop viral proteins, but not the host function of the cell, and creating drugs which mutant strains will not become resistant to. It's a constant battle. The retrovirus is a virus made from RNA, and a prion is going to be an infectious protein. So prions cause a group of rare fatal brain diseases in humans and other mammals. When studying animals with certain neurodegenerative disorders, researchers exposed them to radiation which would have killed DNA or RNA. And it suggested that this infectious agent was a protein instead of a DNA or an RNA, which would have been a nucleic acid. So this is what a normal protein looks like. You can have it be unfolded or folded. It just depends on how compact that protein is. How a prion works. It's going to induce abnormal folding patterns in normal proteins. Healthy people normally eat the meat with the prion, and then it travels through the bloodstream and it spreads throughout the body, settling mainly in the brain. Common diseases: Kuru, which is spread by cannibalism; Scrappy, which infects sheep, that's a typo, and pigs, causing itching; and mad cow disease, which infects cows, and it also leads to them having a loss of coordination, and then they end up falling over. Post-mortem examination of the brain is going to reveal substantial destruction of brain tissue. So that is it as far as our notes are concerned. We're going to go through and do some Google questions. I have a couple HIV videos I want to show tomorrow, and then we have a little virus project to work on. Alrighty, that is all I have for you today. Make sure you turn in that Google Doc of those notes that you took that were in blue, and you will get credit on that. Have a great day.